let's tackle the first question, that stem cells are your repair system. I could go through all the science, but you know today that that's really what is happening. So here's a short video that will describe this process. So the video will focus on the heart, but all this data is coming on models about heart attack, stroke, bone fracture, cut to the skin, burn to the skin. Anytime something is happening, the affected area will release compounds. GCSF, granulocyte colony stimulating factor, stem cell factor, interleukin 8. These compounds, well known today, will go to your bone marrow and will kick stem cells out of the bone marrow, trigger the release of stem cells from the bone marrow. Within 3 to 5 days after an injury, we can quantify in the blood an increase of about 3 to 10-fold in the number of circulating stem cells. These stem cells circulate everywhere. They don't know when they're released where to go. So as they circulate everywhere, the affected tissue is releasing locally one specific compound called stromal-derived factor 1. Your stem cells as a receptor, CXCR4, specific for that molecule. When they connect, the stem cell expresses adhesion molecule, grab the capillary, and basically penetrate into the tissue, migrate in the tissue. When the stem cell gets in contact with cellular debris of that tissue, every single organ and tissue in your body has proteins on the surface of the cell specific to that tissue. So these will basically trigger a signal to the genome, and then these stem cells will start proliferating and transforming into cells of that tissue. It is your repair system. Every single time you've had an injury since the day you're born, that's what took place. We simply did not know. And what really allowed us to really discover it, it's not in that presentation, but it's an interesting tidbits of fact. What really allowed us to develop that is the discovery of a protein called green fluorescent protein. It's extracted from a jellyfish in the 60s. It's 40 years of development. In 2008, it was a Nobel Prize in chemistry. And you just shed light on it and it starts to glow. So you put GCSF in a stem cell, and then you can observe that stem cells migrating in the liver, for example, becoming a population of liver cells. You cannot tell that this was a stem cell the day before, but if you shed a light on it, it starts to glow. Otherwise, the phenomenon is invisible. You can see a stem cell migrating in the tissue, and in about 8 hours, it has started to become cells of that tissue. When you look at it using pathology, you just have a liver cell. No indication that it was a stem cell the day before. So it revealed the whole phenomenon in a way that now is absolutely undeniable. So in all this process, you release stem cells, they circulate, they migrate in the tissue, proliferate, differentiate. The part that has been the most studied and where we have the most information is the link between the number of stem cells in circulation and what it means for health. So let's focus on that.